Okay, great. So welcome to the, I guess it's the 9th lab. Um, so yeah, and so today the main topic that I'm going to be covering for my, I guess, recorded video is Maestro, which is part 1 of the homework. It's kind of it's only for graduate students, part 1 of the homework. So it'll be most applicable to this group. And then I'll also talk a little bit about part 4 of the homework, which is integrating the single cell RNA-seq data with the single cell ATAC-seq data. That's partially graduate, but there are some undergraduate parts as well. And so everybody will probably benefit from that. I don't expect these slides to take, I guess, much more than 30 minutes. So I'll have a lot of time at the end, I guess, to answer more specific questions about the homework that you might have. Yeah, so first I just want to give a little bit of an introduction to Maestro, which is the software that, or the pipeline that you're using in part 1 and kind of throughout the assignment. And so this was actually published by Professor Liu in her group just last year. So this is really kind of at the forefront of research on bioinformatics. And so it's exciting that we get to use it at this point in the course. And so I guess, what is Maestro? Well, basically it's a comprehensive pipeline for the analysis and integration of either single cell RNA-seq or single cell ATAC-seq data. And basically the way that you can think about it is, basically it does everything from the point where you get a FASTQ file all the way until the point where you want to have a process Seurat object. Seurat is the software that Martin talked about last week that's kind of used extensively throughout this homework. So yeah, so basically Maestro will take your raw data and really create a nice, beautiful Seurat object for you to use for whatever kind of analysis that you'd like. So, okay, yeah. And so this is a figure that I took from the paper, and it kind of gives a nice overview of the many things that Maestro will do. And that's also why, you know, if you've tried to run part 1, you'll notice that it takes hours because it's doing so many different things. So looking over here at the single cell RNA-seq half of the figure, I guess. So you can start with a FASTQ file that does quality control. You can filter out cells that, well, I should say from your FASTQ, it first performs alignment and demultiplexes like the barcodes, the UMIs. And then it performs quality control, removes cells that you know, don't have lots of reads or cells where not many genes are expressed. It builds your cell gene expression matrix. It'll perform clustering, a differential expression analysis, and then a very interesting feature is that it will actually annotate cell types for you automatically. So for example, it will, if you have an immune cell data set, it might tell you these cells are B cells, these cells are CD8, these cells are CD4 T cells. And so that's a really interesting feature. Over on the single cell ATAC-seq side, it also does similar things, I guess, from the FASTQ to the point where you cluster, call differentially accessible peaks, and so on. And then one feature is that they developed, I guess, a regulatory potential model. We're going to talk about that a little bit later. But that, I guess, basically allows you to kind of predict which genes are likely to be expressed just from the open chromatin regions. And this is kind of what allows you to ultimately integrate the single cell RNA-seq with the single cell ATAC-seq data. And that's the point of part 4 of the homework. Yeah, so any quick questions about, I guess, kind of the overview of what Maestro does? I guess you'll kind of get to see all of these in the homework. but anything in particular right now? I can't see the chat, by the way. Okay. So now let's talk a little bit about how to actually run this on the PSET or the homework. So it's built using SnakeMake, which is basically a tool to, I guess, design pipelines, right? So Maestro is basically, right, it's going to do things like STAR, RSAM, quality control. And so it's, I guess, each step might be something that already exists. And then SnakeMake kind of gives you a way to kind of combine these all into a single pipeline to run. And so I think on the homework, you've already been given the code. Just part of the challenge is, submitting this as a batch job. I know there's been some questions already about that. And I just want to go over the steps that you should do to run this on Cannon. And so each time before calling the Maestro command, any sort of Maestro command, you need to make sure that you first source the Conda environment. I mean, and the exact code to do that's given in the homework. Once you do that, you want to configure the pipeline using either Maestro single cell RNA initialize or Maestro single cell attack initialize. This only takes a few seconds and you only need to do it once. So for example, you don't need to start a compute node to run either of these two commands. Basically what it's going to do is it's just going to create a metadata file, a YAML file in the specific folder that basically says, right, when it's actually time to run, here's what you're supposed to do. And then once you've done that, then you can actually run the pipeline. Since it's going to take 3 to 6 hours to do both of them, you really need to submit it as a batch job. So this means that you should be using the sbatch command. You should write a shell script that kind of contains the code that you want to execute, and submit that using sbatch. I wouldn't recommend srun, although it could be possible. But for a job this long, you really probably want to use sbatch. I think the parameters that I set specifically for each was 60 gigabytes of RAM, 5 hours of runtime. That's like the time max, and then 16 CPUs. All right, and once you do that, all you need to do is I think you need to copy and paste a couple text files and download a figure to your local computer. Any questions about this in particular? I think afterwards I can show an example of sbatch if some people are a little bit confused about that.
Hey, Philip, I have a question regarding the Conda environment.
Sure.
Is the code provided already included like Lisa in it? Because I think I run into issues with that.
Yeah, I see. Yeah, so it should. It should. Yeah, so, and you're writing like an sbatch script?
Yeah.
Are you sourcing, so like inside your sbatch, inside your shell script, do you source the environment?
Yes, I did.
I see. And I saw this on the Slack, and you double check that it's like there's no typos in the Lisa directory. Wait, sorry, you did double check?
Yeah, I did.
I see. Has anybody else encountered that problem? Okay, so I'll take that as a no. So I guess once I'm done with my slides, if you mind sticking around, maybe you can share your screen. I can take a closer look. Maybe that's probably the best way.
Yeah, sounds good. Thank you.
Yeah, yeah. But yeah, because there's a ton of things going on. There's a lot of potential for something to kind of not work. But hopefully we can all get this ran this week, because you definitely don't want to run this Sunday morning when the PSET or when the homework is due, because it does take many hours. So we'll try to get all these Maestro errors ironed out this week. Okay. Great. Yeah. And so, the code's already given to you. But I just wanted to, I guess, kind of briefly go through, I guess, what kind of ingredients you're providing Maestro when you call single cell RNA-seq in it, because it's probably useful for kind of understanding what the pipeline is doing. And so in your homework, you're going to specify 10x genomics, because the data that we're using was generated with 10x genomics. But like we've seen, there's other platforms for generating single cell RNA-seq data. So Maestro also works with DropSeq or SmartSeq, and those are just different protocols. So the analysis would have to be slightly different. You're also providing, the reference genome, because Maestro is also going to align to the genome, perform quantification with RC. Oh no, that's for QC. RSAM, yeah, RSAM down here. Yeah, that's quantification. Let's see. Whitelist. Does anybody know what a whitelist is, or what the whitelist is used for? Yeah, so the whitelist is, I guess, a list of the barcodes that 10x Genomics uses. Because if you remember from a lecture, I think it was with Martin was giving the lecture, I guess while you're doing PCR or whatnot, sometimes an error, or there could be a sequencing error when you're reading, I guess, the barcode. And so there's an algorithm, I think it's Star Solo, that kind of, even if two barcodes differ maybe in one nucleotide, they can kind of realize that this may have been a mistake, right? Because you're trying to reduce, I guess, or you're trying to find all reads from the same barcode. And so the whitelist is a list of the barcodes that 10x Genomics actually uses, which can correct for these kind of sequencing errors. Yeah. And let's see. So here's single cell ATAC-seq. It's, I guess, similar in nature. 10x Genomics also does single cell ATAC-seq. Yes. Okay. So let's see. Here are a few guidelines, I guess, that you might need to do to run this on Canon. And so first is obviously you need to make sure that you specify the correct number of cores and the correct amount of memory, otherwise it's not going to work. Also, and I think everybody will need to do this, you only have 100 gigabytes of space in your home directory. And apparently your previous homeworks, which I didn't do, use up a large majority of that. So much to the point that if you haven't deleted some of your large files from previous homeworks, Maestro may not run. So do be mindful of that. I think you probably need 30, 40 gigabytes of scratch space to run the pipeline. So make sure that you're deleting your old index file. Like if you have an genome index file from like the previous homeworks, I'd just go ahead and delete that so you have enough space to run Maestro. Yeah, and also, when you call SnakeMake, it's going to look for that metadata file, that YAML file. So you should make sure that like you're in the correct folder that actually has the metadata file when you submit your job. Right. Okay, and so once you've actually ran this pipeline, the questions are pretty simple. I think they just ask you to copy a few text files and download a few images. One is the results of the quality control. And so, here's actually kind of what you're supposed to get. One of them's for single cell ATAC-seq, one of them's for single cell RNA-seq. Does anybody know which one's which? Quick question. Which one is single cell RNA-seq, left or right?
One on the left.
Yeah, yep, because of genes. Yeah, so here you'd be looking more, when you're looking at open chromatin regions in single cell ATAC-seq. So yeah, this is RNA-seq, this is ATAC-seq. Great. And I think probably this is the one that you're looking for. I think they ask you to download the ATAC-seq one. Great. Okay, so if there's any more general questions about part one, I can take that now. Otherwise, I'm going to start talking about part four a little bit. Okay. Okay, so I'm going to talk about integrating single cell RNA-seq and single cell ATAC-seq. And I think this was probably the topic of today's lecture that got rescheduled. And so maybe if what I explained here is a little unclear, it might be best to wait until Thursday's lecture. But I'll try to give you, I guess, the main ideas. So basically, what we can do now is, take our sample and then run both single cell RNA-seq and single cell ATAC-seq. And so we get these two data sets. And one of the questions is, how can we integrate these two data sets in a way that we can kind of get the strengths of both RNA-seq and ATAC-seq? And so, like, for example, one of the main benefits of single cell RNA-seq is that it's very good for cell type annotation. So like if you can see all the genes that are expressed in a cell, then you really have a good idea of what kind of cell type that is. If you're just looking at the open chromatin regions, it can be, you can distinguish between very different cell types, but you might not be really able to distinguish between two kind of similar cell types. So single cell RNA-seq is kind of the best for cell type annotation. And so one of the questions is, can we use the RNA-seq to kind of help annotate our single cell ATAC-seq data if we collected the cells from the same sample? And so in part four of the homework, you're going to use this idea called label transferring to integrate the single cell ATAC-seq with the single cell RNA-seq. And so this figure kind of, you can ignore immunophenotyping and spatial transcriptomics, but just looking at gene expression and chromatin accessibility. So basically, what you're going to do here is, from the single cell ATAC-seq, you can kind of predict which genes are likely to be expressed. And actually, Maestro kind of builds a model which, if there are a lot of open chromatin regions right around, I guess, the transcription start site, then it's kind of likely that this gene is expressed. So you have some information about gene expression from the single cell ATAC-seq. And Maestro, I guess, kind of infers a score for each gene. And then what you do is you treat both kind of this inferred score and your true single cell RNA-seq data as kind of two batches from the same experiment. So up here, the reference might be your single cell RNA-seq data, and the query might be the, I guess, the scores from, I guess, the scores that you inferred from single cell ATAC-seq. And then kind of what this method does is it kind of finds anchors between the two data sets. Well, so actually first you do batch correction, but after that you kind of find anchors between the two data sets. So if, I guess, two cells or if two clusters look very similar between the query and the reference, it's kind of likely that they align and they have the same, for example, the same cell type. And so this is the idea that you're going to use on part 4, which is basically, so like if on the single cell RNA-seq, your reference, you know what type of cells all these clusters are, you can kind of just pass these to the single cell ATAC-seq. And then that's basically what you're going to do. Yeah, and I'd also recommend going to this vignette right here. It's also linked on the homework, but the big hint is that it contains most of the code that you'll need to run this part of the homework. And I think I have another slide about this. Yeah, so what's on the left here basically is that from the single cell ATAC-seq, you can kind of predict the gene activity, or you can predict, you can kind of predict whether or not this gene is likely to be expressed just by looking at the open chromatin regions. And then you kind of get a measurement for each gene, which you can treat as kind of fake gene expression. And from that, you do batch correction between the real gene expression and I guess the fake gene expression. And then, so for example, the blue might be single cell RNA-seq and the red might be single cell ATAC-seq. But if you know the cell type labels for the blue, then you can maybe do something like K nearest neighbors for the red to annotate cell types for the single cell ATAC-seq, which is, like we said, kind of more difficult. So this is a little bit complicated. How can I explain this better, or is this making sense, I guess, what the goal of this is? Right, annotating cell types is a little bit difficult in single cell ATAC-seq. So the goal of this method is to use the single cell RNA-seq to kind of help us annotate cell types. Does any part of this need more explaining? Okay, so I'll move on. Yeah, so I guess the key steps that you're going to perform in part 4 basically is, first you're going to match barcodes between single cell RNA-seq and single cell ATAC-seq. A lot of the code for this is already given, and kind of the rationale behind this is explained in that part of the problem. But then the second part, you're going to run the SURAT function, find transfer anchors, and I guess the anchors are kind of the mapping between the single cell RNA-seq and the single cell ATAC-seq data. And once you run that, you can then, I guess, transfer the data. So by running transfer data to actually annotate the single cell ATAC-seq data using the single cell RNA-seq data. And then the final step, only for graduate students, you're going to see how well this method, this label transferring method did, because you've matched barcodes in step 1, and you, I guess, technically know the true cell types from single cell RNA-seq. You can, I guess, use that as the ground truth to measure how well this label transferring method did. And so that's, I guess, the 4 key steps that you're going to do in part 4. Yeah, and so yeah, in part 3.3, you'll have to make a heat map, which basically, shows you how well your predicted cell type was compared to the ground truth. And the ground truth here is the RNA-seq data. This isn't the actual data set that you guys have, but I'm taking this from the vignette where they used a different data set, but did a similar, I guess, experiment. And so you kind of want to see something like this. This would be evidence that the label transferring method worked well, because, almost 100% of the time, or I guess 80-ish% of the time, if you predicted, for example, a CD14 monocyte, CD14 positive monocyte, that's what the true ground truth label was. And so this is just judging the accuracy of that label transferring method. And also, it also returns like here's how confident I am in this labeling. And then you can compare, I guess, this would be the prediction score, basically, how confident I am in the label. So like, if it's 1, I guess I'm very confident that I know the cell type. If it's low, I'm not very confident. And you can kind of compare the prediction scores between cell types that I got wrong, which would make sense why it's kind of left skewed, and cell types that I got right, which would be more, or I guess would be higher on the right side. Yeah. Any questions about what's expected here? And so like the hint I'm giving, I guess, also is that like, if you want the code to generate figures like this, you should go reference the vignette that I linked. It's also linked in the homework. Okay, so final slide here. I just wanted to give, I guess, a few hints, corrections, suggestions for the homework. In part 1, problem 2, there is a typo. I may correct it and repost it. We do want the cell filtering plot instead of the redistribution plot. So it should look like this. That's the cell filtering plot. The redistribution plot is like a bar graph showing like what percentage of reads are uniquely mappable. You shouldn't give that. Instead, give this. Okay, also, if you're stuck on part 2, problem 2, a hint would be to consider the Seurat function percentage feature set and to look at the provided example. This is the problem where you're trying to look at the fraction of mitochondrial reads in each cell, I think. Yeah, so that's the hint. Look at this function, and their example shows how to look at, I guess, reads from mitochondrial genes. And finally, there's a hint in part 2, problem 5.1, which essentially goes to a GitHub issue page. Just know that, so I think it's like a forum where somebody asks a question and then people answer. Just know that the first answer that the person gave was wrong. And later in the forum, they explain why that person was wrong. Make sure you don't make that mistake, and you read the follow-up, like read the following posts in that forum to understand why the first answer provided was wrong. So just be careful about that if you're using that hint in part 2, problem 5.1. Is any other questions for the video?